Good day, students! Welcome to this new topic about nucleic acid hybridization. This recorded discussion is divided into two parts. The first part will focus on the overview of nucleic acid hybridization, and the second part will include southern blot hybridization, which is one of the few hybridization formats at present. The other formats will be discussed in another pre-recorded lecture. So, to begin my discussion, nucleic acid hybridization is defined as the interaction between two single-stranded nucleic acid molecules to form a duplex or double-stranded molecule. Nucleic acid hybridization takes advantage of the complementary base pairing of nucleic acids. The base pairing of nucleic acids may occur between complementary strands of DNA, between DNA and RNA, and between complementary strands of RNA. That is why it is important for you to learn the base pairing rules by heart. So as a review, always remember that adenine base pairs with thymine and cytosine base pairs with guanine. However, for RNA, there is a slight modification to this rule. Take note, however, that in nucleic acid hybridization, the two participating populations of nucleic acids should be single-stranded. Furthermore, the hybrid formed as a result of nucleic acid hybridization can be unstable or stable depending on the extent to which complementary base pairing takes place between the two strands. As a result, the double-stranded nucleic acid molecule formed can be partially or completely double-stranded. So if there is a high degree of complementary base pairing between the two strands, the hybrid formed is stable. Moving on, the hybridization assay is composed of the basic components. These are the probe, the sample, the controlled conditions that allow for complementary base pairing, and lastly, the controls. So the description of each will be described in the succeeding slides. The first basic component of a nucleic acid hybridization assay is the probe, which is defined as a labeled molecule that binds specifically to the molecule of interest. Probes are often produced through recombinant technology. The molecule of interest here would either be your target DNA or RNA sequence. The probes help in the detection process and determine the specificity of the hybridization reaction. Another basic component of the hybridization assay is the sample. This can either be an unknown DNA or RNA. Since we take advantage of the reversible and sequence-specific binding of the two strands of nucleic acids, then it is important that we have an intact sample at the beginning of the hybridization assay. This is the reason why maintaining the sample integrity is of vital importance in nucleic acid hybridization assays. This can be done by employing proper and standardized sample processing techniques. An example of this would be the immediate snap freezing and/or the addition of lysis buffers to preserve the target. Also, once the target has been extracted, it needs to be purified before the hybridization proper to remove inhibitors and maximize the accessibility of the target probe. The next basic component of hybridization assays is the set of controlled conditions that permit the complementary base pairing of nucleic acids. The sensitivity and specificity of the hybridization reaction are greatly influenced by the physical-chemical environment during the reaction and subsequent detection or collection of hybrid molecules. In practice, the hybridization cocktail is the medium used to control the environment of the hybridization reaction. Empirically designed, the hybridization cocktail is a mixture of reagents selected to favor the interaction of nucleic acids through sequence-specific hydrogen bonds rather than via charge. Just like any other biochemical reaction done in vitro, Nucleic acid hybridization also requires a set of controls for validation purposes. These are the positive and negative sample controls. The positive sample control is known to have the sequences that are complementary to the probe, while the negative sample control does not, and is thus used to monitor the specificity of the assay. So those are the 4 basic components of a nucleic acid hybridization assay. Now these are the most common nucleic acid hybridization assay formats that are currently employed in research and diagnostic laboratories. These are southern hybridization, northern hybridization, in situ hybridization, and microarrays. In the next slides, I will be talking about southern hybridization. And as mentioned earlier, the other formats will be discussed in another pre-recorded lecture. This is the second part of my discussion today. In this part, I will be talking about the first nucleic acid hybridization format, which is southern hybridization. Southern hybridization, or otherwise known as southern blot, is a hybridization technique that allows the detection of a given DNA sequence in a complex mixture of DNA sequences. It can be used to identify homologous sequences in genomic DNA. The technique was named after Mr. Edwin Southern, who was the first person to report the procedure. Now, let me explain the overview of the steps involved in southern hybridization or southern blot. Southern blot starts when a purified DNA sample is cut with restriction enzymes to generate multiple fragments. The fragments are separated according to size through agarose gel electrophoresis. Then, a series of steps are performed to prepare the fragmented DNA for blotting or transfer. Once the fragments from the agarose gel have been primed, these are then transferred or blotted to nitrocellulose. In the final steps of the procedure, the DNA fragments are exposed to a labeled probe that is complementary to the region of interest. The first step in southern blot procedure is digestion of test DNA with restriction enzymes. 10 to 50 micrograms of high-quality or intact genomic DNA is used for each restriction enzyme digestion for southern blot analysis. This concentration may vary depending on the sensitivity of the detection system and the abundance of the target DNA. Digestion is carried out for an extended time to allow complete cutting of all sites in the DNA sample. High specific activity enzymes are also used to ensure complete cutting of every site. Incomplete cutting will result in anomalous patterns, complicating interpretation of the southern blot data. The fragments resulting from the restriction digestions are resolved by gel electrophoresis. After electrophoresis, it is important to observe the cut data. The figure shows a 0.7% agarose gel stained with ethidium bromide and illuminated by ultraviolet light. Genomic DNA cut with restriction enzymes should produce a smear representing billions of fragments of all sizes released by enzyme digestion. The goal of southern blot procedure is to analyze a specific region of the sampled DNA. To achieve optimal hydrogen bonding between the probe and its complementary sequence in the resolved sample DNA, the double-stranded DNA fragments in the gel must be separated into single strands and transferred to a nitrocellulose membrane. One preparation procedure done on resolved DNA in the gel is denaturation. This is the process of separating the DNA fragments into single strands. This can be done by exposing the gel to a strong base, such as sodium hydroxide. However, one limitation of this procedure is that it cannot denature DNA fragments that are greater than 500 base pairs in size. For larger fragments, they first need to undergo depurination in dilute hydrochloric acid before denaturation in sodium hydroxide. The dilute hydrochloric acid removes the purine bases and loosens up the larger fragments for a more effective denaturation process. Before exposing the denatured sample DNA to the probe, the DNA is transferred or blotted to a solid substrate that will facilitate probe binding and signal detection. This substrate is usually nitrocellulose, nylon, or cellulose. There are 3 blotting techniques commonly used at present. These are capillary transfer, electrophoretic transfer, and vacuum transfer. I will first discuss about capillary transfer. Driven by capillary movement of buffer from the soaked paper to the dry paper, denatured DNA moves from the gel to the membrane. The DNA will adhere to the membrane, which will subsequently be exposed to the probe. Capillary transfer is simple, inexpensive, and can be performed in the absence of any instrument. However, this technique is slow and can give less optimal transfer when using large gels. The next blotting technique is electrophoretic transfer. This technique uses electric current to move the DNA from the gel to the membrane. This system utilizes electrodes that are attached to membranes above and below the gel. The current carries the DNA transversely from the gel to the membrane. And then we have vacuum transfer. This blotting technique uses suction to move the DNA from the gel to the membrane in a recirculating buffer. Like electrophoretic transfer, this method transfers the DNA more rapidly than capillary transfer in 2 to 3 hours rather than overnight. Also, it avoids discontinuous transfer due to air trap between the membrane and the gel. However, one major disadvantage of this technique is that it requires expensive equipment and maintenance. After the transfer of the gel-separated DNA to a solid membrane support and hybridization or binding of a specific probe to the target sequence of interest, the next step is to detect whether the probe has bound to the immobilized target. The probes to be used can be radioactive phosphorus atoms. The signal of the probe is detected to indicate the presence or absence of the sequence in question. The original method entailed hybridization of a radioactively labeled probe to detect the DNA region to be analyzed. So that's it for the overview of nucleic acid hybridization and southern blot hybridization. The discussion about the remaining hybridization formats will be uploaded soon. Thanks and have a good day.